Alright champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days. And today we are going to talk about cancer. We are going to end this chapter really soon. We just have one more topic left, that is drug and alcohol abuse. After that, the chapter is done and then we would be having the mock test. And I was thinking I would be giving you a small revision for the whole chapter if possible. Okay, I'll see to it. If it's possible, then I'll give it for sure. Now, going ahead, let's talk about cancer. So when I was a student, I used to study about cancer and AIDS, about which we studied in the previous lecture. All of this gave me a sense of already being a doctor somehow, because now we are studying about diseases and we could just show it off in front of our relatives and all when they ask certain questions or when they are not aware about certain things. And when we speak about that, it gives a sense of pride. So obviously knowledge in any ways is really good, and yes, we should be appreciating it. And also, as I said, we spoke about AIDS in the previous lecture. If you want to have the PDF for the same or any other lectures that we already covered, then children, you can join the WhatsApp channel. You can get the PDF there. Yeah. Now, were you able to solve this question? Well, two of my students who were there, they were able to solve the questions and they also put down in the comment section. Inshara and Sukanya, I'm really happy that you did that effort to put down the answers in the comment section as well. It's a great thing. I feel really happy when I see all these comments. Now, HIV enters which T cells? You know, HIV is a virus which is the causative agent for what, children? It is the causative agent for AIDS, acquired immunodeficiency syndrome. Right. Now, what is a T cells that gets affected with HIV? So, as I said that day also, there are too many different type of T cells, but we have to be mainly focused on TH cells. And if at all you ever forget, just understand HIV starts with an H and T helper cells also has a H. Okay, so the helper T cells that get mostly affected, and when the number reduces, that's when our system also become very weak. Okay, now second question, death in AIDS is due to what? Aggravated immune response. Is it because we have too much amount of immune response? Due to opportunist infection, that is, so usually what happens is that our body is able to combat all the diseases that comes through the way. Okay, so there might be so many opportunistic, bacteria, virus and fungi which sneaks inside our body whenever our immune system is really less. So is it because of those opportunistic infections, due to inflammation of genitalia, that is inflammation of genital organs, or due to diarrhea and fever? Now, death in AIDS, they are talking about death. They are not asking about the symptoms that is being shown. So, the initial symptoms that people have is fever and diarrhea and all this thing. Okay, that happens after a long time. And then they also acquire a lot of diseases, small diseases which they could have overcome if their immune response was really good. So, here death is caused because of the opportunistic infections which comes along our way. And since our immune system is really weak, they utilize this opportunity to cause what? Death. So, it is B. Okay. I hope you understood that. Now, if you have any doubt regarding AIDS, you should be seeing that particular video. So, the number of questions for this particular chapter has been increasing, as I am telling from day one. So, we cannot skip this chapter. It was giving 4 questions the previous year, so 4 into 3 is 12 marks. Sorry, 4 into 4 is 16 marks. You cannot lose 16 marks just because of any factor that is there. We have to study, and this chapter is really interesting for the future doctors as well. Now, let's begin to talk about cancer, and since you know how I teach, first we have this thing over here. We have to fill the difference between the cancer cell and the normal cell, depending on what's there in this particular paragraph. Okay, so let's start one by one over here. Cancer is one of the most dreaded diseases of human beings and is a major cause of death all over the globe. So, it is a major cause of death, remember that. More than 1 million Indians suffer from cancer and a large number of them die from it annually. We know that, it's a very sad thing. The mechanisms that underlie development of cancer are oncogenic transformation of cells. Its treatment and control have been some of the most intense area of research and still going on. In our body, cell growth and differentiation is highly controlled. Very important thing. What do you mean by that? Basically, the cell, okay, for example, this is one cell and says I want to grow so big. Can it grow so big because just it wants to? No, because there will be certain things in our body which will be saying, no, you are not allowed to do so. There are certain checkpoints, there are certain control checkpoints which does not allow our cells to grow rapidly or differentiate into something else that is not destined to be. Okay, so there is a control system in our body. Now, in cancer cells, there is breakdown of these regulatory mechanisms. So, basically, cancer cells on the other hand, they are like, chuck it, we do not want to listen to you. We are in an old world, we would grow, we would divide, whatever we want to do, we would be doing it. That means initially the proteins which were controlling the cell growth, they are also hijacked. They are also blocked. Their work is also blocked in a way that these guys, the cancer guys, the cells, they do their own will. Okay, normal cells show a property called contact inhibition, by virtue of which contact with other cells inhibit their uncontrolled growth. So, for example, just imagine, okay, so since contact inhibition is the first thing, I will write it over in the next slide. Okay, so what is it saying? What do you mean by contact inhibition? It is when contact with other cells inhibit their uncontrolled growth. Basically what happens, this is a normal cell and this is our first point, that is contact inhibition, how it is different over here and here. So, if I put a normal cell or maybe a bunch of normal cells over here and ask them to divide. Okay, so what happens, basically they keep on dividing and it's all good. But once these cells come in contact with other cells which are dividing, this particular process of division, it stops. Since they are coming in contact with each other. Since because of this contact, what is happening, they are not going to divide again. This is what you mean by contact inhibition. You are inhibiting someone or something because you are getting in contact. On the other hand, when you talk about cancer cells, these are naughty cells. Basically, what are they going to do? They are not going to listen to anything that is happening in our body. So, for example, just imagine I am making red. Cancer cells are the red cells, okay? And our normal cells are the yellow ones. Basically, normal cells comes over here, touches it and it stops dividing. But do you think cancer cells are like that? Will they follow this particular rule of contact inhibition? Never. They are not going to do that. So these cells keeps on dividing and forms a lump there. Okay, what does it form? It forms a lump there. So they do not follow contact inhibition, whereas they definitely follow what? Contact inhibition, children. Okay, next thing, let's see what is there. By virtue of which, okay, cancer cells appear to have lost this particular property. As a result of this, cancer cells just continue to divide, giving rise to masses of cells known as what? Tumors. So this particular mass of cells that you see over here, what do you call these mass of cells, children? You call these mass of cells as what? Tumor. Basically there is difference between cancer and tumor. Understand that. Okay. So, one cell, it is there. It is dividing uncontrollably and it is forming a bunch of cells. Now, this whole mass of cells, what will you call it? You will call it tumor. Okay. Now, these cells can again divide, can again go into different areas and cause trouble. That is when we say that cancer has been caused. If the tumor remains like this, we have a different name for it. So, there are different type of tumors. One is the one that can spread around. The other one is the one that does not spread, remains at one place, does not cause big trouble. Okay. So, you will come across that, but please understand, tumor, the word tumor means a mass of cells together which has been growing because of uncontrollable growth. They grew like that, but there are different type of tumors. One type of tumor is cancerous, whereas the other is not cancerous. Please understand that. We'll come to that, okay? Now, when I'm saying one type of tumor cells is cancerous, with that I am going to talk about the next thing, which is known as metastasis. What do you mean by metastasis? Metastasis means basically the cells Okay, for example, we have cells over here, our normal cells. These won't be moving around to different places, going at different places for picnic and be staying around there. No. But our cancer cells over here, they are not like that. They basically, for example, this is a blood vessel, then this is an organ or something like that. They basically move around. So, sorry for making it in white. Basically, the cancer cells, they move around from one place to another, cause trouble there, then they will move to the next place, cause trouble there. This process is known as metastasis. What do you call it? Metastasis. And this is a very important feature of cancer cells. They are the ones which moves around, cause division there, moves around, cause division there, which is why they have a rapid spread. We say no, the cancer cells have spread it throughout the body. It is because of this particular feature which is known as metastasis. Does our normal cells do it? Okay, our normal cells are not able to do it. Okay, going ahead, we have proto-oncogenes. It's not pro-oncogenes, it is proto-oncogenes. Children, just give me a second, just a second, okay? Yeah. So, basically, as I said, it is, okay, proto-oncogenes. So, what are proto-oncogenes? Generally, in our body, you know that we have a cell, we have a nucleus. Inside the nucleus, what do we have, children? We have our DNA, right? Inside the nucleus, we have our DNA, and DNA has lot of different segments which are known as genes. These segments that you see over here, which is known as genes, and each gene is responsible for formation of some protein. So basically our DNA is made up of small fragments known as genes, and these genes are responsible for making some of the other proteins. So there are certain genes that is known as proto-oncogenes. Proto-oncogenes. So whenever they are normal, whenever they are there, we have them in our normal cells also. We have these proto-oncogenes. So I am marking this in red. Okay, we have proto-oncogenes in our normal cells also, and they are responsible for producing normal proteins over there. They are responsible for producing what? Normal proteins over there. But over here in cancer cells, what happened? Basically they are going to do some mutation in this. So if this was our DNA, so how is cancer being caused? So cancer is being caused because suppose the proto-oncogene was there, but here this proto-oncogene has some changes in it. Okay, has some changes in it, some mutations in it. Okay, some mutations in it, which is why now this particular cell is going to produce some protein which could be responsible for uncontrolled growth of the cell. It can cause uncontrolled growth of the cell. Basically, I am trying to say that this particular activation of this particular proto-oncogene, or basically some changes in the proto-oncogene, will shift from normal function to a very altered function, which could cause what? Cancer as well. So better I think I should not have written it over here. This is how you can get cancer. Okay, this can be a cause of cancer. This particular thing can be under cause of cancer. Okay, but yes, you should be knowing that definitely in our cells there are genes which are known as proto-oncogenes. You will be studying about it next in the next topic as well. Okay, so there are proto-oncogenes which does normal function of producing normal proteins, but due to some alteration in these genes, some mutation because of some what say UV rays, something like that, basically now it is going to produce some other protein, or probably it was not producing some protein, but now it has started producing some protein which is lethal for the cell. Okay, it could be either way. Certain things could happen, but anyways, whatever it is producing, it is not really good for our cell. That's the thing that you need to remember. Okay, now apart from contact inhibition, which was the first thing, then we have second thing, that is metastasis. The third thing that the cancer cell shows is telomerase activity. What do you mean by telomerase? So telomerase is an enzyme. Okay, telomerase is an enzyme. So if you know about chromosomes, no children? If you know about chromosome, the end region of the chromosome, no? The end region of the chromosome, it is known as telomere. Okay, it is known as telomere. And you already know one thing, that a normal cell, it undergoes some set of division and then it stops dividing. The cell aging has happened. Now it's time to die. The cell won't be dividing again and again. So basically, why does this happen? So it happens because every time during cell division, every time during cell division DNA is being copied and things like that happens, right. But it does not copy the last particular portion, okay, because it is not efficient. Our enzymes are not that efficient. So basically at this point of time, every time this is unreaded, this particular portion is unreaded, and every time our DNA or basically our chromosome is going to get shorter. During each cell division, our DNA or basically our chromosome is going to get shorter, and at the end it will obviously stop dividing. It is obviously going to stop dividing because the chromosome size is decreasing. Fine, but can it be done in cancer cells? Cancer cells are those which are immortal. They are not going to die anytime soon. They are what, children? They are immortal. So they cannot be dead. They cannot be dead. So what mechanism do they have? They say that we have an enzyme, we can activate the enzyme, that is telomerase, okay, which could basically maintain the length of telomere. Which could maintain the length of telomere such that during each and every cycle, the length of the telomere, it remains the same. It remains the same. There won't be change in the telomere size, and the cell can keep on dividing. So normal cells, don't they have telomerase? They definitely have the enzyme telomerase, but the activity is kind of low or absent. In our case, in normal cells, children, the activity of telomerase enzyme, it is low or absent. Basically, it is considered to be low because that is a natural mechanism. We want the cells to die and new cells to form, because the old cells obviously could have more sets of damage, and we do not want that. Our body is very efficient in clearing out all these damaged pieces, okay, more than us. So basically, similarly over here also same thing happens, but they say that here we want the telomerase enzyme to be very active. So telomerase enzyme becomes very active such that telomere region is maintained even after each cell division, such that these cells remain what? Immortal, children. Okay, they remain immortal. So we have contact inhibition, we have metastasis, we have telomerase activity. Now, since I am going to exclude this point, I am going to add up one new point for you guys, and that is angiogenesis. So fourth point will be angiogenesis. What do you mean by that? Angiogenesis. When you say angioplasty, angiography, everything has to do something with your blood vessels or your heart and things like that. Similarly, I am going to say something over here. That is, basically, suppose this is a cancer cell, okay, and these cancer cells, obviously they are going to make a lump, they are going to grow around, right? They are going to grow around. So they need constant supply of nutrients. They need constant supply of nutrients. And imagine that there are blood vessels around over here, but it's not reaching their lump of cells. It's going to some other areas, things like that. So what do? They build their own blood vessels. They make mechanisms in which way the blood vessels may be reaching over to them. That is what you mean by angiogenesis. Genesis means formation. You are making new blood vessels for them. So I read it somewhere. So basically you can think of a construction site. So this is the construction site, that is our cancer cells, and in a construction site they take the they take the pipelines and everything from the nearby areas for the time being until construction is done, and then they think of something else, right? So for the initial period they need their own pipelines. So similarly they are making their own pipelines in our body with the help of themselves itself. So cancer cell has this very unique ability to form blood vessels which could supply them enough amount of nutrients, and all of these cancer cells could be really happy with these nutrients. They could survive, they could divide and have a very jingalala life. Okay, so that is one feature about cancer cells. They are also responsible for angiogenesis. Okay, so I'm just summing things up. What is the difference between normal cell and cancer cell? normal cells basically show contact inhibition. What do you mean by that? When they come in contact with other normal cell which is dividing, they stop their division over there. Whereas when you talk about cancer cells, they are like, we are not going to listen to anything, we are just going to divide. And now when they divide, they are going to form a mass of cells which is known as what? Tumor, children. It is known as tumor. Going ahead, what do you mean by metastasis? Metastasis is when these cells are going to migrate to other places in our body, even without our permission. So these cells can go to some organ, from there it can go to some other organs. Well, this particular feature of migrating throughout the body, having a picnic throughout our body, it's not done by normal cells, it's just done by the cancer cells. Okay, because their motto is to just go around, habitat everywhere, take their nutrients and survive. Okay, third feature is telomerase activity. So in our normal cells, there is an enzyme that is known as telomerase, and this enzyme is basically low or it is absent. It does not work efficiently over there because it is not asked to work efficiently. We want our cells to die after some point of division. So basically what happens, at the end of a chromosome, we have a region which is known as telomere, and every time the division happens, the telomere size is get reduced. When obviously it gets reduced, basically you're cutting down the chromosome shorter, right? So at one point of time, there will be a point where it will stop dividing. The whole cell will stop dividing. But over here, we do not want our cells to die. Over here, they want their cells to be immortal, which is why they say telomerase, please help us over here. We need you over here, which is why they maintain the length of telomere, and the whole cycle of cell division continues, and they remain immortal. Now, the fourth point is angiogenesis. That means they are going to form their own blood vessels so that they get enough amount of nutrients and everything that is required for their growth. Am I clear? Am I clear with this? Will you be able to write this if an exam it comes? Yeah. And also proto-oncogenes, we will be talking about them when we talk about the causes of cancer. Okay, now let's talk about tumor. So we have types of tumor, benign and malignant. We are going to read about them now. Tumor are of two types, benign, okay, and malignant. Benign tumors normally remain confined to their original location. That means, are they showing metastasis? No. And do not spread to other parts of the body and cause little damage. They only cause a little amount of damage, probably some inflammation or a lump around in your body. Yeah. The malignant tumors, on the other hand, are a mass of proliferating cells called neoplastic or tumor cells. Okay. So malignant cells are the ones that proliferate really quick. Okay, they are going to divide. And these cells, individual cells, what do you call them? Neoplastic or tumor cells. That means they are like plastic. They can change whatever DNA, whatever they want, and they do it continuously. Okay, these cells grow very rapidly, invading and damaging the surrounding normal tissues. As these cells actively divide and grow, they also starve the normal cells by competing for vital nutrients. Obviously, if you are dividing, for example, you are there and one of your friend is there, and you are eating the food. You have very limited amount of food and you are eating the food continuously. Your friend will be starving. Similarly, over here, they do not consider our normal cells to be friends, but still, they do not provide any nutrient for them. Instead, they take every nutrient. They are very selfish. Okay. Cells slaughtered from such tumors reach distant sites through blood, and wherever they get lodged in the body, they start a new tumor there. I told you. And what do you call this process as? This property is known as metastasis, and it is the most feared property of malignant tumors. Please remember it. It is what? The most feared property of malignant tumor. So with this information, can we just fill up this particular sheet? So it's easy, right? This one is obviously easy. So it's just a lump of cells that do not spread around our body. Okay. So are they going to cause cancer? No. This is the one that causes cancer, and I was talking about this only, that all the tumors are not cancerous. Okay, all the tumors are not cancerous. Just understand that. So this is not a cancerous one. Now, when you talk about malignant term itself means it is going to destroy something. Okay, so malignant tumors are the ones that it spreads throughout the body It spreads throughout the body by what? What do you call that process, children? Metastasis. And while they do that, they go and reside at areas, they starve the normal cells for nutrients. For nutrients. Okay? And what? Metastasis is the most feared property, okay, of cancer, malignant cancer. Then what? Cells of these tumor are known as what, children? Are known as neoplastic cells. Neoplastic cells. Okay, they can quickly bring about changes in their DNA. They are like plastic. We say no, it can be reused, it can be rechanging every time and then. So that is why we say it neoplastic. Okay, so I hope that is done. And malignant one is the one that causes the dreaded disease, that is cancer. Okay, and how do they starve? Because they have the power for angiogenesis, to make new blood vessels around them. Done? Now, next thing, causes of cancer. So, let's read it. Transformation of normal cells into cancerous neoplastic cells may be induced by physical, chemical or biological agents. So, we have three agents: physical ones, the chemical ones and the biological ones. These agents are known as carcinogens. So, all together, the ones that causes cancer, okay, the ones that causes cancer, what do you call them? They are known as carcinogens. Okay, the causative agents of cancer. Going ahead, ionizing radiations like X-ray and gamma rays and non-ionizing radiation like UV cause DNA damage leading to neoplastic transformation. So these are the physical factors, okay, something that happens physically. So there are rays that are going to attack on our cells, our normal cells, and they are going to damage our normal cells. Okay, and how can you remember the name of ionizing radiation and all of them? Let's see that. So ionizing radiations are the ones basically who has the power, no children, they have the power to attack the atom, and break the bonds between them such that the electrons can be released. So ionizing radiations are the powerful ones which basically attacks on the atom and makes the bond broken. Okay, breaks the bond. I am writing like that. Breaks the bond in an atom. And releases what? The electrons and everything. In this way, obviously our cells are going to get weak, and obviously these are free electrons can go and react around things. That is also a problem. So they are okay. So these are about ionizing radiation. And what about non-ionizing ones? They are going to directly affect our DNA. They are going to cause some damage in our DNA, some alteration in our DNA. And this alteration together, what do you call them? You call them mutations. Okay, when there are some changes in our DNA, you call them mutations. Okay, so in our normal cells, mutations are going to happen in our DNA, and DNA is the one that will be controlling what proteins are made. So now some other proteins are made, or some proteins are not made which are required to stop the cell growth. So all this problem could happen. Now what are the examples for ionizing radiation, children? It is X-rays. So we say we should not be taking too many amount of X-rays. Why? When people go to take X-ray, no, we are said that we should not be taking too much amount of X-ray, which is why people are told to be outside. Only the person who is affected has to be inside, so that you don't need to affect the other person as well. Okay, because of the strong rays. But again, we have to use it as a tool to find a lot of diseases, including cancer. So that is one thing. But we are not supposed to be in regular contact with X-rays. Okay, apart from X-rays, we have gamma rays. we have gamma rays. You know what is the story of X-rays? In this way, probably you might remember ionizing radiation as well, because when you associate with some story and all, it might have nothing to do with this, but still. So there was some ray which was, the person who found it was not able to understand what to name it, and which is why he named it X, because usually when maths also you say X plus Y, find the X. So that is something unknown, which is how X-rays the term came. Okay, and okay, fine, anyways, leave it. So X-rays and gamma rays, two of the things are there for ionizing radiation. Now non-ionizing radiation, example will be what? UV radiations. Non-ionizing is one visible light that we have, that is also non-ionizing, but UV is the one that causes lots and lots of trouble. So it is going to cause DNA damage. By how can it cause DNA damage? By causing mutations, some changes in our DNA which could lead to abnormalities. Okay. Now that is the physical factor. So both of them, they are what? They are the physical causative agents of cancer. Now, going ahead, what do we have? Chemical carcinogens present in tobacco smoke have been identified as major cause of lung cancer. Now, children, everyone are advised. You are in this age where you are going to, you have this particular mentality of trying new things out to be cool or something like that. I was also in your age. I know what you guys think, and that's why, and we do not have much age difference as well, which is why I am telling you that basically we should not be trying things that could be lethal for our body. So our body also will say that try it out, try me, then I will also bring out cancer in my body. So that is how things work. How do these chemicals work? So when we say that smoking is bad or injurious to health, in every movie and the starting of the movie, it is said that smoking is injurious to health, but still people smoke. Why? Because basically the tobacco smoke which is being produced during smoking, it has a lots of content of benzene and things like that, which is going to affect your normal cell, again bring out certain mutations in your DNA such that it could give altered responses. Okay, which is why we say that we should not be smoking. Okay, and also, as I said, which is the most common cancer which is caused by tobacco, it will be what? Lung cancer. And I'm not even kidding. I have a brother of mine who is a doctor, okay, and he works as a maxillofacial surgeon. Okay, so for all the surgeries that are present over here, that are done over here. So basically, they do lots and lots of operation, surgery and all for cancer patients, and they have a prehistory of tobacco smoking and also chewing of tobacco. So basically this particular chemical that you see over here, it's very dreadful disease, and whether it's slow, but definitely it could show some effect in your body. If not cancer, then some other disease. So definitely we should be trying to avoid the intake of all this thing. But if you're asked what is the cause of lung cancer, children, it is caused because of tobacco mostly, and it's because of the smoke, because the smoke has benzene and lots of other chemicals, and this tobacco and everything, when the regular smoke gets in, okay, it causes inflammation in your cells. And now you only think in the lung, no, basically the airways, if it is causing inflammation, okay, there is bulge over here. Inflammation means something is happening which is going to pressurize other small cells. They are going to get damaged, and because of this recurrent damage again and again, it is going to destroy your airway also in some or the other way, which is again a problem, and it could also lead to cancer, because there is some abnormality over here and cancer cells love abnormality. They want that. So basically they come in search of that, they just start spreading over there. Okay, so please understand that. Now going ahead, what do we have? We have biological agents. So for biological agents, we have two things. One is viral oncogene and the other one is proto-oncogene. So there are some, okay, we have to read it first and then we'll come. Cancer causing viruses called oncogenic viruses. Oncogenic is the ones that causes cancer. What do you call the doctors who treat cancer, children? Oncologist. Okay, oncologist. So basically cancer causing viruses called oncogenic viruses have genes known as viral oncogenes. What does this viral oncogenes do? If you have studied AIDS with me, you know that basically these viruses they basically come inside our normal cells, okay, and we have our DNA. Our DNA is cleaved, okay, our DNA is cleaved from middle, and then this slowly in between this particular DNA, they establish their DNA. And now when our body will be reading out DNA, it will make proteins for that, right? So basically they are using our machinery to make proteins that they require, and they sustain in our body, and this could be cancerous. So they could make certain proteins which could inactivate certain other proteins which was protecting us. You get it? So always, obviously, there are so many cells, so many proteins involved in our cell cycle, such that some of them stop the growth of the cells. Some of them, they find where the DNA is damaged. If DNA is damaged, that cell is being killed. Okay, there's a natural cell that happens, which is known as apoptosis. Everything of this is controlled. But in a cancer cell, all of this is uncontrolled. So you are making proteins, the cancer cells are making proteins, which would tell other proteins to shut down their activity. So obviously they will work. So these are viral oncogenes. These genes that you see over here, they are viral oncogenes, and they are put over here with the help of oncogenic viruses. Okay. Going ahead with talking about proto-oncogenes, I already told you, in normal cells we have certain areas and certain genes. You call them proto-oncogenes, which are producing normal proteins in our body. They produce normal proteins in our body, okay. In normal condition, they are going to produce normal proteins. Now, when altered or when mutated, when this particular gene is mutated, basically what will happen? There will be certain abnormalities in the cell, because there will be certain proteins which will be produced which we do not need. Okay, so these are known as proto-oncogenes, which are already there in our body. Okay, dear children, so this is the normal causes of cancer. These are the normal causes of cancer: physical, chemical and biological. I hope you got it. Please remember, X-ray and gamma ray comes under ionizing radiation. Or you can just remember, if you go to X-ray room, people are not advised to come near it because it is dangerous. And why is it dangerous? Because it has to be ionizing. Okay, non-ionizing are the ones UV rays. We say that UV causes skin cancer and all, right? Because of this thing. Because UV rays, if you have it in a lot amount, and if your skin and your body is prone to it, then probably it could affect you really easily. Chemicals, tobacco, because of the smoke, it has benzene and a lot of other chemicals which could cause mutations in our DNA. Yeah, and all these things. Done? Going ahead, diagnosis of cancer. Now that is important. Chalo. Early detection of cancer is essential as it allows the disease to be treated successfully. So obviously early detection is important, you know that. Cancer detection is based on biopsy and histopathological studies. What do you mean by biopsy and histopathological study of the tissue and the bone marrow? Histopathological studies of the tissue and blood and bone marrow test for increased cell counts in case of leukemia. So this is given in your book. What do you mean by leukemia? Basically, when cancer is going to affect your blood cells, okay, white blood cells, basically that, it is known as leukemia. What do you call it? Leukemia. What do you mean by biopsy? That is the first question that we have. Biopsy is when, for example, some X-person has cancer. Some X-person has cancer and they have a lump. This particular lump, okay, some part of it, this is a lump, some part of it is taken. This is known as biopsy. Now, when tests are being done, okay, when this particular cells are being examined, they are being studied, you call them histopathological study. Histo as in what? The term histo means tissue. The term histo means tissue. So when you are studying tissue, to check whether they are uncontrollably growing or things like that is happening. They are moving, they are immortal, they are not dying. That means probably this particular tissue is a part of a tumor which is malignant, which could be cancerous. So that is what you mean by biopsy and histopathological study. I hope you understood that. Okay, going ahead. Okay, and one more thing, what tissues are you going to search for? Basically the blood, okay, basically blood and the bone marrow also. For leukemia, I'm talking about leukemia. If it is leukemia, what do you have to look for? If it is leukemia, then blood and bone marrow. Why bone marrow? Because bone marrow is the area where the blood cells are continuously dividing. Okay, blood cells are being formed in the bone marrow, which is why we look for cancer over here, cancer cells over here. Okay, and also in the blood. Now leukemia, as I said, is for blood. Now talking about computer tomography and radiography. So in radiography and computer tomography, I'll tell you one thing, X-rays are being used. X-rays are being used to see through our body, okay, to check whether there is some deformities over there. Now computer tomography, or what you call as CT scan, gives you a 3D image of the same. That means something that has more clarity. But when you talk about radiography, just talking about X-rays, and that is going to give you not 2D. It is going to give you a 2D image of the same. Okay, so this one would be more reliable for having a better understanding of things. This is what you mean by computed tomography. Computed tomography, CT scan. X-rays are only used in both these, okay, but 3D image is gotten over here, whereas 2D is gotten over there. Fine. Now computer tomography, I wrote it over there. Okay. I know there is a mistake. Okay. Now we have MRI. Very important. Very important, but we didn't read the other one. So let's read it first and then we'll go ahead, okay. So in biopsy, a piece of suspected tissue cut into thin section is stained and examined under microscope. And this studying is known as histopathological study. So biopsy is basically cutting a thin piece of tissue which is there, as I showed you, and now when you study it, you call it histopathological studies. Techniques like radiography, which use X-rays, CT scan and MRI, magnetic resonance imaging, are very useful to detect cancer of the internal organs. So internal organs, obviously, for that you cannot go around cutting and doing things initially. Okay, which is actually been done. Biopsy has always been done initially, but still you have technologies like MRI and CT and radiography for getting things done as well. And what happens in computer tomography and everything, let's read that also. Computer tomography uses X-ray itself to generate a 3-dimensional image of the internals of an organ, I told you, of an object. MRI, now that's something new. MRI uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Okay, what is being done over here in MRI? What does MRI stand for? Magnetic resonance imaging. So over here, magnetic field is utilized to find any deformities. Okay, and also one more thing, you saw over here, what is X-ray, children? A type of ionizing radiation. A type of ionizing radiation. Now over here in magnetic resonance imaging, what are you going to use? Non-ionizing radiation. Non-ionizing radiation, which is why we say that this is the safest, it's a very important point, the safest way to diagnose, which is why I am putting 3 stars. It is the safest way to diagnose. What is it? MRI, where magnetic field is used and non-ionizing radiations are being used, which are going to cause least damage to our body. Okay, that is one thing. Now what about antibodies? What about antibodies? You know how antibodies work. Basically, there is an antibody, okay, and suppose there is an antigen. It has the perfect site to bind this particular antigen, then both of them will combine. Then only both of them will combine. If they do not match, they won't be combining. So basically over here, what is being done? From a person who is susceptible to cancer, who is probably suspected to have cancer, not susceptible to, who has cancer, who is thought to have cancer, from them antigens are being taken. Antigens taken from patients, and now it is allowed to treat with certain antibodies that we already have. So we know that these antibodies are produced by our body during cancer. So we have these antibodies overall. And now, if we see that these antibodies are binding with this particular antigen that is being taken from the patient, we will get to know that okay, since it is binding to an antibody which is being released in our body when cancer is there, then probably this particular person will be having what? Cancer. That is how antibodies work. So over here, antigen is taken from the patient's body and then it is made to react with antibodies. Okay, so it can be done in different ways. I'm just giving you an idea. It's not like you have to take in out and things like that. It has a huge different mechanism. I'm not going in detail, but at least how it works. Okay, the basic thing. Now, this antibodies can be linked to some enzymes or something, such that it could show that this particular linkage has taken place. Okay, it could be linked to some enzymes. You studied this in 8th as well. So this is the total overall mechanism of how things work in this particular detection method. We'll read it once. Okay, antibodies against cancer specific antigens are also used for detection of certain cancers. Techniques, okay, that is also done. So there's a very short thing that is given in your book. I just wanted to give you a brief explanation, that's all. Now, techniques of molecular biology can be applied to detect genes in an individuals with inherited susceptibility to certain cancer. So, we know people who are having breast cancer, it is said there is high probability that their children could also have breast cancer. So, in this case, there are certain techniques in molecular biology where it is going to examine your DNA, your genes and already pre-check whether you are susceptible to cancer. So those techniques are also available, but I'll say it's still developing in our country. Okay, so basically we have biopsy and histopathological technique, CT scan, radiography, MRI and then antibodies, 5 of them in total. Just have a quick read, it's a very small thing. Okay. Now, last but not the least, the common approaches for treatment of cancer are surgery, radiation therapy and immunotherapy. Okay, we have surgery, we have radiation therapy and immunotherapy. In radiotherapy, tumor cells are irradiated lethally. What happens? Basically, the tumor cells, for example, these are the tumor cells and they are radiated, okay, such that these cells would die. Why would they die? Because already they have lots of DNA damage. Already they have DNA damage and their DNA repairing technique is poor. And their DNA repair technique is what? Poor. So basically in radiotherapy, if this is radiotherapy, it is going to target fast dividing cells. Fast dividing, I'm messing it around, just a second. Fast dividing cells. Now these are targeted, and if it is cancerous, obviously they will be having DNA damage. Okay, because they do not take care of it. Our normal cells, they definitely do take care of it. They have DNA repair system as well. And their DNA damage also cannot be repaired since they have a poor DNA repair technique as well. So obviously they will die. But these radiations, if they fall on our normal cells, we definitely have DNA repair techniques to make it clear. But it is made sure that our normal cells are being protected from this particular thing. But obviously, because it's a radiation, it could affect the other cells. So which is why radiation therapy is given in breaks. such that during this particular break our normal cell could repair. Okay, that is about radiation therapy. Now if you talk about surgery, what happens in surgery, you already know about that. Basically, if again, as I said, this particular person is having cancer and there is a lump being formed, this lump is being removed with the help of with the help of surgery. So that is what you mean by surgery. Radiation therapy, I told you, it is done in breaks, and targets rapidly dividing cells with poor DNA repair strategies. Okay. Now what do you mean by chemotherapy? Chemotherapy is when basically you are giving chemicals, and these chemicals again are going to target cells which are rapidly dividing uncontrollably. Okay, and what are these cells? Basically your cancer cells. But what happens? You might have seen your hair starts to fall during chemotherapy. Why? Because the hair follicles are also rapidly dividing cells. Okay, and also you might be seeing the immune cells, their amount will also get lowered because of the same reason. Okay, so chemotherapy is basically going to affect the cells. We are giving chemicals in the form of medicines and things like that. Basically at this point of time, the rapidly dividing cells are being targeted, which is basically cancer cells, so they also die, but along with it, sometimes they also die. Now there are techniques such that the hair follicles and all is not being targeted, but yeah, it is very costly. Talking about immunotherapy, what do you do? Since you might have seen in virus, no? In virus, what do they do? For example, if one cell is affected, it gives out interferons. It produces interferons, and these interferons are signals which tells the other cell that you have to be protected. Please obviously release some antiviral agents so that you could be protected. That is the thing. So in immunotherapy, we are giving alpha interferons. The doctors gave alpha interferons, and these interferons, no children, will be making other cells aware, and they will be ready for an immune response. They will make the immunity go a bit stronger. That is the meaning of this thing. So basically, if I try to divide, what do I get? Okay, radiotherapy, tumor cells are irradiated lethally, taking proper care of the normal tissues surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors, obviously. Majority of drugs have side effects like hair loss and anemia. Okay, anemia, why do they have anemia? Because over here, immune cells, basically your RBC, okay, so your, okay, why, okay, your RBC, everything is also dividing in your bone marrow, and they also die, which is why anemia is also caused. So apart from hair loss, basically anemia is also caused because of the same reason. Okay, so yeah, hair loss and anemia. Most cancers are treated by combination of surgery, radiotherapy and chemotherapy. That is, three of the three things are being utilized for treating cancer mostly. Tumor cells have been shown to avoid detection and destruction by immune system. So usually our tumor cells would be going and hiding somewhere so that they do not be detected by the immune system as well. Therefore, the patients are given substances like biological response modifiers such as alpha interferons, which activates their immune system and help in destroying the tumor. I just told you that interferons, alpha interferons, please remember alpha interferons. The interferons there is different. Alpha interferons are given such that they could elicit the immune response, and our body itself can fight against these diseases, because our body obviously will be in a very, very weak position. So that's about surgery. That's about the treatment of cancer. And with this, we have to solve the previous year questions. So let's solve it one by one. This is one question. You can try to pause the video and answer it. This is the next one. Okay. Again, pause the video, answer it. Next one. Next one. Next one. Okay. So basically, you can give it as 1, 2, 3, 4 and 5. Please answer these questions in the answer box, chat box or in the comment section. Okay, I want to see your answers, then only go ahead with my answers. So now it's time to solve my answers. Well, if you are reached here, you didn't attempt for yourself, please try to attempt it once by yourself and then listen to me. Okay, now which one of the following is not the property of cancerous cells, whereas the remaining 3 are? So they are asking you which is not the property of cancer cell. They compete with normal cells for vital nutrients. They definitely do. They do not remain confined in the area of formation. No, they do not. Exactly. They do metastasis. They divide in an uncontrolled manner. Exactly. They show contact inhibition. Totally wrong. Do they show contact inhibition? Do they stop when other cell is there? No. So basically that's wrong. Carcinoma, what do you mean by carcinoma? Something out of the box? It refers to malignant tumors of connective tissue, malignant tumors of skin and mucus, malignant tumors of colon. Basically it refers to malignant tumors of skin and or mucous membrane. Carcinoma means the cancer cells which are going to affect the epithelial tissue in some or the other way. Epithelial as in the tissues which are going to cover your organs or your bodily structures, everything. So skin and the mucous membrane is one such thing. Okay. Now the blood cancer is known as, tell me children, we studied about it. The blood cancer is known as leukemia. It's a very easy question, right? Now cancerous cells can be easily destroyed by radiations due to why? What is the thing? How can it be destroyed with the help of radiation? Because of what? Rapid cell division, fast mutations. What could be the reason? You tell me. So shall I leave this question for you? I think I should be leaving this question for you and solving the homework question, because this is an interesting question, whereas the homework question is comparatively easy. So please do this as homework. This is your homework question. Okay. Cancer cells can be easily destroyed by radiations due to why? What is the reason? Which one of the following techniques is safest for detection of cancers? I told you the safest one is magnetic resonance imaging. That is the safest thing to be done. Okay? Because in this way you are not cutting your body part or doing anything. It's just all okay. So people get confused with histopathological studies and this, but this is the safest way. Okay? Because you are using non-ionizing radiation. So please remember, this is the homework, not that. And one question for you, this I will solve. Which of the, or you can also try solving it in the comment section. Which of the following statement is not true for cancer in relation to mutations? Mutations inactivate the cell control. Mutations inhibit production of telomerase. Okay, it is asking what is not true for cancer cells in relation to mutation. Mutations inactivate the cell control, it is correct. Mutations inhibit production of telomerase, is it correct? Because I think telomerase is needed. Mutations in proto-oncogenes accelerate the cell cycle, yes, obviously. Mutations destroy telomerase inhibitor, it is also correct. So the correct answer is what? Mutation inhibits production of telomerase. Mutation won't be inhibiting telomerase production, it will be definitely saying that telomerase you have to be produced because we need it. Okay, again I am telling you the homework is this question because I found this question to be more tricky now while I taught you. Okay, so that's all children. I hope that you understood what I tried to teach you. And if you like this video, do not forget to share and subscribe. And also one more thing, please give your 100% while you are studying. Go behind, go beyond your NCERT. Try to look for other questions as well. Not be just limited to these questions. Even though this will definitely give you a boost of confidence, which I want. And be happy and rest assured everything is going to be really, really good. Thank you. Have a really, really nice day.